When it comes to genetic disorders, the general public and scientists alike normally have a lot of unanswered questions. For example, how does DNA determine traits? There's a lot of unpacking to be done in order to answer this. First, we must discuss transcription, translation, and mRNA processing. Transcription is a process that occurs in the nucleus of the cell using RNA, a copy of DNA, with bases adenine, guanine, cytosine, and uracil. It is used to code and create proteins and instructions for the cell. First, initiation occurs. This is the beginning of transcription. It begins when the enzyme RNA polymerase binds to a region of a gene called the promoter. This is a region of DNA where RNA polymerase binds in order to initiate transcription. The term start point refers to the position on DNA corresponding to the first base on RNA. A terminator is a sequence of DNA that causes RNA polymerase to stop transcription. Termination is the ending of transcription and occurs when RNA polymerase crosses a stop codon in the gene. The mRNA strand is complete and it detaches from DNA. Translation is the process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after transcription. The ribosome decodes by starting the binding of complementary tRNA anticodon sequences to mRNA codons. Transcription and translation take place in different parts of the cell. Transcription takes place in the membrane-bound nucleus, whereas translation takes place outside the nucleus in the cytoplasm. A codon is a sequence of 3 nucleotides on an mRNA strand that depicts a specific amino acid. 3 codons are called stop codons, which tell the cell to stop translating the mRNA and to not encode an amino acid. For most amino acids, multiple codons exist, each of which could code for that amino acid. Each codon specifies a particular amino acid in the polypeptide chain. A peptide bond is a chemical bond formed between 2 molecules when the carboxyl group of one molecule reacts with the amino group of the other molecule, releasing a molecule of water. This is a dehydration synthesis reaction, or a condensation reaction, and usually occurs between 2 amino acids. Professionals use codon tables to display what each codon means and how it tells the cell what to do. mRNA processing is also a pretty important part of how cells function and carry out specific tasks. In the nucleus, pre-mRNA is produced through transcription of a region of DNA from a linear chromosome. This transcription must go through processing, or splicing and addition of the 5' cap and poly A tail while it's still in the nucleus. This helps it then become mature. Introns and exons are nucleotide sequences within a gene. Introns are removed by RNA splicing as RNA matures, so they're not expressed in the final messenger RNA, while exons go to be covalently bonded to create mature mRNA. The 5' cap protects the mRNA from degrading and assists in ribosome binding during translation. The poly A tail aids in the export of the mature mRNA to the cytoplasm and is crucial in binding proteins involved in initiating translation. Now let's get into the specifics of retinitis pigmentosa, a genetic disorder having to do with the eyes and vision. Retinitis pigmentosa, or RP, is the name given to a group of inherited eye diseases that affect the retina, the light-sensitive part of the eye. RP, as it is more commonly known, causes the breakdown of photoreceptor cells, which detect light. Photoreceptor cells capture and process light for us to be able to see. As these cells break down and die, patients experience progressive vision loss. Fewer than 200,000 cases are diagnosed per year. It begins with the loss of night vision. Night blindness is when you cannot see in the dark. Your vision may be normal during the day. When you start losing night vision, it takes longer to visually adjust to the darkness. You may trip over things or have trouble driving. You also might find it harder to see in dark places like movie theaters. Next, many experience a gradual loss of peripheral or side vision. This tunnel vision can lead to bumping into new things as you move around. This is because you're not able to see objects below and around you. Some people also have problems with central vision. This can make it hard to do things like read or thread a needle. Some people may also have trouble seeing different colors. There's a a variety of general testing available for this specific disease. Genetic testing looks like a sample of blood or other tissues to see if you have certain genes that are associated with a disease. It can also help determine the likelihood and severity of a disease and whether gene therapy might be helpful. Electroretinography is a test that measures the electrical activity of the retina, or how well the retina reacts to light. It works kind of like an EKG, which measures the activity and health of the heart. Visual field testing for retinitis pigmentosa can measure your side vision and find blind spots. An optical coherence tomography, or OCT, is an imaging test that captures special, highly detailed pictures of your retina. It can help diagnose RP and uncover the specifics about how it's affecting your retina. Retinitis pigmentosa, or RP, is a highly heterogeneous genetic disease, including autosomal recessive AR, autosomal dominant AD, and X-linked inheritance. Recently, ARRP has been associated with mutations of EYS, or eyes shut homolog, which is a major causative gene for this disease. The normal function of the G protein transducin is to assist in phototransduction in rods and cones of vertebrates. This gene is located on the human chromosome 3. It contains 47 amino acids and it is expressed more often in photoreceptor cells. The most common feature of all forms of RP is a gradual breakdown of rods and cones, which detect and process light and color. Symptoms of RP are most often recognized in children, adolescents, and young adults, with progression of the disease continuing throughout the individual's life. The pattern and degree of visual loss are variable. Retinitis pigmentosa is an inherited disorder and therefore not caused by injury, infection, or any other external or environmental factors. People suffering from RP are born with the disorder already programmed into their cells. Doctors can see the first signs of retinitis pigmentosa in affected children as early as age 10. There is currently no cure for RP, and there are a few various treatments, but not all of them work. Research is being conducted in areas such as gene therapy, transplants, and retinal prosthesis. Since RP is usually the result of a defective gene, gene therapy has become widely explored for future research. Electrical stimulation, radiation, and decoder chips are other possible options.